and then we will get started. All right, there we go. And then we will get started. And then we will get started. Okay. Welcome to the Earth to Earth seminar. Thank you for joining us today on the 8th of April, 2021. As a reminder, the mission of Earth to Earth is to engage the UK geosciences community with inspiring science from internationally leading researchers, to promote inclusivity and diversity, support early career researchers, and highlight geoscientists' contribution to addressing global environmental challenges. It's open to all UK universities for participation. And today's lecture will be recorded, and we have speaker permission to make it available on the YouTube channel with closed captioning. Calendar links and further information are on our website at earth2earthseminar.com. For questions, please use the Q&A channel on the bottom of your Zoom window. You can post these anytime during the talks, and I'll have a look at those. We should have about 10 minutes at the end for the speakers to address your questions. So now it's my great pleasure to introduce today's speakers. Today, we're fortunate to be joined by Professor Liane Benning and Dr. Stefanie Lutz. Professor Benning is a biogeochemist and head of the Interface Geochemistry Group at the German Research Center for Geosciences in Potsdam. Professor Benning's group combines experimental and field-based approaches with state-of-the-art analytical methods to explore microbe-mineral-fluid interactions and their impact on the global cycling of elements. And Dr. Lutz is a microbiologist and ecologist currently based at Genexa in Switzerland. Dr. Lutz's research interests are in the diversity of life and its ability to survive and thrive in extreme environments. Really exciting stuff. So today they will be talking collectively about the contribution of microbes and minerals to melting the Greenland ice sheet. And I will pass it on to.
Thank you very much. Aubrey. And first of all, I would like to thank the organizers for inviting us to give this talk. And this journey that we started started about almost 10 years ago with Steffi and I in Yorkshire, for all of places in the UK, from which we both then moved on. And we talk about a little bit of how we actually studied microbes and minerals to try to understand a global process, how it changes. And so if you think about, I started going to the Arctic about 30 years ago, probably more years than many of the audience in terms of age, but I observed in those 30 years quite dramatic changes, which are probably obviously linked to the increase in temperature. The melting of the ice and the changes that you see in the Arctic are actually accelerated in comparison to many other places in the world. Now, as a geologist, this is not a surprise in a way, because on the long term, we see these kind of fluctuations all the time. We saw them through the geologic history. Whether we look at the sort of snowball Earth in the Proterozoic, or we had like hot houses and cold houses throughout all the geologic history. So these things happen. We have these alternating cycles, which have gone all the time, happen naturally. However, when we now look back at the Phanerozoic, this is the last 500 million years, we saw that the CO2 in the atmosphere at the beginning was much, much higher. The variation was much higher, and only with the onset of the land plants, the gymnosperms and the angiosperms, both in the Carboniferous, and then later on, we have actually the drawdown of CO2 to sort of the levels of roughly today. This coincides with actually these glacial periods and the hot periods in the geologic history. So again, at long time scales, these variations are not surprising. If we now take it a step back and just go to 1 million years, that means we come closer and closer to today. When we look at the epic ice core, we see that the partial pressure of CO2 and methane over 1 million years has fluctuated. Again, we fluctuate, for example, CO2 between, let's say, 200 and 250 ppm, and methane between, let's say, 400 and about 650 ppm. But basically what we had at the present, we have a very dramatic increase and very fast increase of this thing compared to everything before. Showing you about 1 million years, but this is now going back a couple of million years already. If we look forward, the future is very scary because that's the kind of prediction that we're having, and we don't know how and what the triggers are. If we now bring it back to 20 million years, that means we're coming back to the last glacial maximum. What you see here on the left-hand side, you see the North American continent melting and Antarctica over the last 26 million years. So you see it starts on the left-hand side with quite a lot of ice, which disappears dramatically and only remains at the end just Greenland. And in Antarctica, the melting also happened, but much less. If you transform this now into a two-dimensional plot to see how much water is delivered to the oceans through this melting, in this 10K year period, you actually have at the 4 to 5 degree temperature increase, and the sea level rise was 130 meters. The majority of 130 meters came from the North American continent, but also small amounts from Greenland, Fennoscandia, and Antarctica. But the ice loss at the time, cumulatively, was 3,000 gigatons per year, which is a huge number, but still remember that number, because we'll come back to it. I downloaded this morning from the Mauna Loa measurements, because already now, compared to the average that we had in the last million years between 200 and 275, we are already at 418. And so basically, these are the last 3 decades that I went to the Arctic with. And you see that since the Industrial Revolution, actually we have an accelerating increase of these values in terms of the CO2 in our atmosphere. Now, obviously, this is linked to a temperature increase. And this is a wonderful heat map from Ed Hawkins, where you see on the y-axis, you see years, on the x-axis, you see months. And you see that the temperature increase is dramatically going up. And in November 2015, we have globally reached above 1 degree pre-industrial level. This is translated obviously in amount of water, which is in terms of the sea level rise. And this is a nice presentation from Kevin Pluck, where basically showed that in 140 years between 1818 and 2017, we added only in those 140 years, we added 22 centimeters of water to the global sea level rise. So that's actually quite a lot of water. If we then think of what happens in Greenland, if all of Greenland would melt, and we're not talking of it melting like that, we're talking it melting over decades, centuries, we could add up to 6 to 7 meters of fast. If we now look really to more like today, what you see here is that 2002 to 2014, accelerating the sea level rise in the Greenland Ice Sheet. You'll see very often the GRIS label, which is Greenland Ice Sheet. And only in these 10 years, we're actually adding more than 1 millimeter per year to sea level. And this mass loss is accelerating. And this is developed from the gray satellite measurements, where they measured mass loss in total. Now, if we have globally reached 1 degree pre-industrial level, in Greenland, we are already at 1.8 degrees, and we are approaching a critical state, a tipping point. But you remember that in the 10K years of the last glacial maximum, we had reached, with the massive disappearance of the North American ice shield, we have reached 10 times this value. And here we now reach the 250 gigatons per year already in very, very short period of time. The consequence is actually due to the fact that the cumulative melt days on Greenland have changed. For example, between 1979 and 2007, we had on average about 50 melt days per year across Greenland. Only a decade later, this is 2012. Yes, 2012 was an exceptional year, but since then, almost every year seems to be the exception. We had at one point in June, not just higher temperatures in Greenland than we had in the whole of Europe, but we had 97% of Greenland melting. And the average days at the moment seems to be more like 75 days per year of melting, not 25. And obviously that changes, because that means in the periphery of Greenland, more and more of the snow and the ice are melting. Why is this all important? Is because actually to predict the Greenland Ice Sheet surface mass balance, SMB, for a model in the future, let's say up to 2300, is very difficult. Because this is the 1 degree at 1 millimeter per year which we have in 2015, and you see that the predictions, depending on what you take, can be really wild. The biggest problem in these calculations are the albedo, because that's the biggest uncertainty. Albedo is measured it's a number measured from satellites. When you look from a satellite, and this is the way Greenland looks like, you actually see in the western margin very dramatically, sort of like a dark band, but this dark band you see also in small pockets in the rest of Greenland, but in the western margin, it's very dramatic. So what actually does that mean? If you now zoom in, and we are now looking at this western margin part here, we see that actually this is a bare ice zone where you see the snow line, and then at the moment you have already in the dark zone comprised of lots of lakes, but also this very, very dark band. And in the next couple of slides, I'll just zoom into this. So basically now you're down at 1 kilometer, you see the lakes, and you see quite dirty ice. You go down to 50-meter scale bar here, you see the rivers, and you see some patches of white, but actually ice is not what most people think of pretty ice, but it's actually dirty ice. And if you now bring it down to 10 centimeters, you see that even at these small scales, you have huge differences in the way the structure of the ice looks like and the darkening, so from very clean to super dirty. So what we're trying to figure out is how to actually, what causes that and what is the effect of it. When people measure albedo from satellites, they saw that, let's say between 81 and 2000, this is a value, it doesn't matter, just look at the color, it's just green. 10 years later, it already dramatically changed. This is just pure satellite measurements. So they try to explain this, and they say, well, okay, the first is snow and ice physics. Everybody knows that if you have wet snow, it's darker than fluffy snow. In terms of albedo, this dry to wet transition, if you think albedo as 1, explains at best down to 0.75. But this is insufficient to explain the changes that we've seen from the satellite. The second parameter is obviously black carbon. And I'm using black carbon as a communal name here for anything which comes from wildfire or from fossil fuel burning, anthropogenic fossil fuel burning. And this is the only parameter that is at the moment included in global models, in long-term global models. But it's based on optical properties of the snow in the spring. We know nothing about its chemical reactivity. We know nothing of its fate during the summer. But we also know that together, the black carbon and the wet and dry are still insufficient to explain the albedo measurements we have. And logically, the third thing would be mineral dust. And you'll see from now on calling, and I see very often use the word MD. Whether this dust comes from the Sahara, or it comes from the area where the glaciers retreat and they leave glacier flour in front of it, and then it gets picked up in the Arctic desert and put back on a glacier. Only in 2014 has been suggested to actually try to include these. But it's really difficult because we don't know what it is. We don't know where it comes from. We don't know what the fate in summer is. We don't know what the albedo reduction is. So if we just take these three together, mineral dust, black carbon, and snow and ice physics, that means wet and dry, we actually don't know what the different parameters do. However, most people, when they go to Greenland or they think about Greenland, they think of a nice white surface, but actually very often at the end of the summer, this is the way that it looks like. And the reason why it looks like this is because it changes dramatically. If you now look at the scale bar here across my image, the scale bar here is 5 meters, you see that this dark surface, 10, 15 years ago, people just thought this is dirt, but actually it is biology. And in the peripheries, you see, let's say, red snow or green snow. So these are snow. This is not ice cream. This is straw. This is red, green snow, and red snow. And on the ice, you see these purple, dark brownish colors, which actually all color. They are pigmented algae, which color these environments. They are not included in the surface mass balance models, but they are the ones which during the summer, during the melt, change the fastest because they grow fastest. But we don't know. When we started, it was very little known. It had just started to be described that they are there, but we didn't know who they are, what they're doing. And so basically, what we need to know is if we want to understand what the light absorbing particles do to albedo, we have to take all of them. We have to take what they do from when they come from the air, what happens when they are in the snow, what happens when they're in the ice. And the 3 parameters which we'll touch very briefly is black carbon, Then I'll pass over to Steffi to tell us everything about the microbes, and I'll finish up with the mineral dust. The problem is everybody just measures once. So with the projects that we have been running for the last couple of years and running in the future, we try to measure all the parameters together to actually get a better picture, because if you just measure once, you miss the other. So we need to measure them individually, combined, and specifically the difficulty is to do it during a melting season. Only by doing this can we upscale and then get a value to include in the predictive models. So I'll just show you a little bit about the black carbon. And this is work from Janine McCutcheon and my colleague Jim McQuaid and Steffi. We've done this as part of the Black and Blue, where we collected everything from the air, because we need to know what comes from the air before it actually develops. And we collected all kinds of parameters, and I'll just show you one example. This is the way the black carbon looks like. So note the scale bar here is now 5 nanometers to about 300 nanometers. So these little aggregates, they are extremely black, so they obviously affect the albedo in the snow, but we know nothing what happens to them. The reason why it's important to know is because depending on how they combusted, whether they are fossil fuel or whether there's a tree burning in Canada, their light absorbing properties change. And that means we can actually measure that with a variety of techniques, spectroscopic techniques, to figure out what their effect on albedo is. At the moment, this is work in progress, is driven also by Janine McCutcheon. And the second big part is obviously the pigments from the algae. This is what I now pass over to Steffi, which tells us everything about the algae.
Thanks, Liane. So, yeah, Liane has already briefly introduced the biology, and indeed glacial surfaces are full of life, and they've been recognized as one of Earth's biomes. A plethora of organisms can be found on glacial surfaces, including bacteria, algae, fungi, nematodes, tardigrades, insects, and also every now and then even humans. And here I would like to focus on the main primary producers and briefly introduce them and their habitats, starting with the snow ecosystem. The primary producers here are the snow algae that Liane has already mentioned. And after the onset of melting in spring, when liquid water becomes available, snow can turn into green, red, or yellow snow, depending on the algal pigmentation. Green snow is caused by young flagellated snow algae, and with increasing irradiation, they produce protective pigments, carotenoids, which act as a sunscreen. It's not fully understood yet what triggers this transition from green snow to red snow. And also not all green snow turns red. So there's still a lot to study. And also green snow is a more short-lived transient stage, and red snow is much more widespread and can be found more often. Less common is also yellow snow, which is caused by a completely different group of algae, the so-called golden algae. And with ongoing melting, the snow cover disappears and the bare ice surface is exposed. And there we can get ice algal blooms, which cause a grayish, brownish darkening of the surface because of their purpurogallin pigment. And by far less is known about those algae compared to the snow algae, probably because they've been overlooked for a very long time, because it really just looks like some inorganic dust on the ice surface and is much less striking than the green or red coloration of the snow algae. And then there's also cryoconite holes that form when particles accumulate, and due to local heating and melting, there are these holes. They're often filled with liquid water, and the primary producers here are bacteria. Here you can see a schematic of a glacial ecosystem with the blooms and the habitats I've just described. And at the beginning of the melt season, those snow algal blooms are more dominant. And with increasing melting, the snow line moves up, and also the snow algal blooms move up. And more bare ice gets exposed, and the ice algal blooms become dominant. And these changes can happen very, very quickly. And Liane already mentioned the extreme melt event in 2012. That's also when we went to southeast of Greenland, to Mittibad Glacier, where we followed these changes in habitats over a 3-week period. When we got there in the beginning of the melt season, the whole glacier surface was still covered by clean snow. And after a few days of melting, some green snow appeared, and some of it turned red with increasing melting. The snow line moved higher up. We got the ice algae blooms at the bottom, and then towards the end, the ice algae became dominant. And all glacial microbes, not only algae, have to cope with a multitude of stress factors on glacial surface, including desiccation, nutrient deficiencies, higher radiation, especially between the snow and ice grains because light's more reflected, constant freeze-thaw cycles, which also lead to osmotic stress, and also that determines also the nutrient availability, where the nutrients are dissolved and available. That's why these algae are polyextremophiles and makes them interesting to study for a wide range of fundamental cellular processes, but also, as Liane already mentioned, because of the albedo reduction potential. And I only want to briefly mention this here. Due to the algal pigmentation, the surface is darkened, therefore less light is reflected, more heat is retained, and therefore more melting appears. And Liane will be talking about this more later on. Our research is driven by the questions: who is there? Looking at the community composition, what are they doing in terms of functions, and how do they affect the environment in terms of albedo and melting? And we are pursuing a multidisciplinary approach using various omics techniques, including amplicon shotgun metagenomics, metabolomics, and also metatranscriptomics. But here it's in bracket because this is work in progress and will not be covered in this talk. We combine these also with targeted metabolite analyses, including pigments and fatty acids, and also a wide range of physical chemical parameters, for example, nutrients and trace metals. And when Liane and I started working on glaciers, our main focus was on the snow. So most of the data I will be presenting will be on the snow, and later I will briefly make the transition to the ice. In order to answer the question, what controls, or what is the community composition and what parameters control the community composition, we did a large biogeography study of red snow across the Arctic. It shows 4 well-separated locations. We chose Svalbard, high up in the Arctic, 6 glaciers at low latitude, at low elevation, with high concentrations of easily leachable elements. Then Tarfala in northern Sweden at high elevations, and closer to proper vegetation like trees. Iceland with completely different bedrock, namely basalt, and with input of volcanic ash and high iron concentrations, and then the glacier in southeast Greenland I've just mentioned. We looked at the eukaryotic community composition using the 18S RNA genes, and if you look at the bar charts, if you just look at the colors, you can see they all look very similar. So all 4 locations have a very similar community composition. Only 6 species made up more than 99% of the community. And then also when you look at the PCA plot on the left, it's not surprising that there's no clear clustering according to the locations, simply because the samples were all so similar. And then again, no surprise that we could not find any correlations for the algal species and any of the quantified geochemical parameters. Suggesting that the community composition is independent of location-specific factors, and most likely physical processes such as melting and availability of liquid water plays a much more important role than the snow chemistry. And this was also suggested by other studies on snow algae. And in contrast to the algae, we got very different results for the bacteria. Here displayed the bacterial classes. If you look at the bar charts, you can already see the differences between the 4 locations. And you can also see some clustering according to the locations. And for the bacteria, we also find correlations with the geochemistry, for example, for sphingobacteria with carbon species, namely dissolved organic carbon and total carbon. Next, we were interested how the geochemistry influences the metabolites. First, we were looking at pigments because those cause the darkening of the surfaces. And here you can see the relative abundance of the different pigment groups. And not surprising, secondary carotenoids make up the largest proportion because those are also that are responsible for the red coloration. They're here on the y-axis, and the x-axis you have the total carbon to nitrogen and carbon to phosphorus ratios. And I've also included the green snow samples here for comparison, and the black line indicates the Redfield ratio for optimal carbon to nitrogen and carbon to phosphorus ratios. And what we found was a correlation between low carbon to nitrogen ratios and higher secondary carotenoid production, suggesting that there's a link between low nitrogen and higher secondary carotenoid production. And we could not find such a link for phosphorus. Then pigments are often stored in lipid bodies, which shield the chloroplast and protect it. And also astaxanthin, one of the main secondary carotenoids, is often esterified with fatty acids. That's why we looked at fatty acids next. Here again, the relative abundance of the different fatty acids and polyunsaturated fatty acids were very abundant. You can see them here again on the y-axis, and again, x-axis, carbon to nitrogen and carbon to phosphorus. And similar to the pigments, we found a correlation between low carbon to nitrogen ratios and higher abundance of polyunsaturated fatty acids, but not for phosphorus. So this suggests that the nitrogen availability seems to play an important role in the metabolism of these algae. Then we were also looking at cell numbers and biomass. And there were small differences between the samples, but overall no significant difference between the different locations. And we could also find a clear link between albedo and biomass here on the x-axis and albedo on the y-axis. And you can see with increasing biomass, there is a decrease in albedo. With this, I would like to move on to a different study in Svalbard, where we used metagenomics and metabolomics to get a better idea of the life cycles of these algae. And here we found these really intensely green and red colored snow patches next to each other. And from the Amplicon data, we knew the community composition, which was very different. And we did some metagenomic analyses. And as you know, metagenomes always contain reads from all organisms present, not just algae, but also bacteria, fungi, and etc. And we tried to pull out all the algal reads, and we were not very successful with this. We're, for example, trying to map them to closely related reference genomes. But the problem is simply that in general, there are not many reference genomes for green algae and definitely not for cryophilic algae. And the closest reference genome is actually Chlamydomonas reinhardti, which is a mesophile and doesn't have any of the cryophilic properties. So at that point, we decided it's important to take a step back and get appropriate reference genomes. So this is in collaboration with Thomas Leyer from the Fraunhofer Institute in Potsdam, who is head of a culture collection of cryophilic algae. And we chose five strains from his culture collection, which are representative for our snow communities. And we did some initial sequencing using short read Illumina sequencing. And what we found that most of the genomes are rather large, and also they're very repetitive, meaning with short read sequencing, we will not get fully resolved genomes. So there's definitely some long read sequencing needed. We already got some PacBio data and also working on some nanopore data. Then we were also doing some metabolomics analyses on these samples. And we could identify quite a few metabolites, you can see on the left, and then we grouped them into different pathways. And for the green snow, what we found is that there was a high abundance of metabolites associated with growth and development, For example, auxine in the tryptophan degradation pathway is a plant hormone which enhances, for example, growth in chlorella. And then what we also got from our targeted carbohydrate data, high abundance of ribose, lactose, which are also part of various molecules involved in proliferation. Then the red snow was characterized by a high abundance of storage metabolites, for example, sphingolipids. They are involved in the hardening of cell surfaces. So when red snow forms spores for overwintering, for example, and also there was a high abundance of the uridine monophosphate biosynthesis involved in synthesis of ribonucleotides, which could serve as storage compounds for overwintering, for example. And of course, this is only a snapshot on 2 samples and 1 time point, but it is a start, and we are currently working on also getting a time series and, of course, more samples. Then, as I mentioned before, I want to briefly make the transition to the ice. And as I already said, there is much less known about these glacial ice algae. They've been described first in 1870 by an Arctic explorer, and then there have been very, very few publications. And only in 2012, which seemed to be a good year for glacial ice algae research, their albedo changing potential has been recognized, and also their pigmentation has been identified. And since then, there have been several publications quantifying the algal-derived albedo reduction. And also Liane will be talking a bit more about our contributions later on. Very briefly, I want to show our initial sequencing of these glacial ice algal communities. Again, using 18S sequencing. And when you just look at the colors, you can see a very, very low algal diversity. We collected several samples across a 100 kilometer transect in southwest Greenland in the dark zone that Liane had already mentioned. And there was one very dominant algal taxon, Ancylonema nordenskiöldi. And with this, it's up to your turn again, Liane. Thank you.
Thank you. So basically, I'll continue up here, and indeed, we have now. Oh, if I can. I hope that with the data that Steffi has showed you, it's clear that actually algae are actually dominating both the snow and the ice domains. And as part of the Black and Bloom project, Chris Williamson has actually done a fantastic study where actually looking at the rate of photosynthesis in the primary production, he actually demonstrated that if you think of just about the ice algal surfaces, they're almost as productive as a eutrophic lake. So obviously this is a least forgotten biome, which has to be taken into consideration when you think about large scale processes. So we now talk very briefly about black carbon and about microbes, but actually we haven't looked at minerals, and that's this darkening is not just black carbon and microbes, but it's also minerals, but they are very poorly understood. So this transect that Steffi mentioned, I will show data of this transect because one of the things you see that if you collect samples from super clean to very dirty, you see these little pockets of sort of aggregated minerals, which if you put them underneath a microscope, they sort of look like that. This is now a scale bar here of 500 nanometers, microns. And these are mineral algae microbe load in one of these environments, because if you get the particle load, those are the kind of data that the modelers need to actually put into their models. So what we then done, and this is work by Janine McCutcheon, where we looked at particle loading, and now the value you see here is nanograms per gram of water, which is basically the amount of melted snow or ice. And we have collected samples as a function of 2 particle sizes, so 0.2 to 5 microns and above 5 microns. And when you take a core, you actually see that in the core, the majority of the particles are relatively small, and only a small fraction is above 5 microns. What comes in from the air and I only showed you black carbon data, but we also have mineralogy data, actually has a larger proportion of bigger ones, but still the total particle size is relatively small. As the snow weathers, the big particles seem to be changing, and if you go from early season clean ice and late season clean ice, you see that the particle accumulates over time. So at the moment, this is just the clean habitats. If we now look at the dirty habitats, where we talk the algal-rich habitats, the first thing you see that overall, the total particle loading is far bigger. Remember, this is a log scale here. And you also see all of a sudden that the distribution between small and large particles is actually changing slightly towards older parts. We want to know what actually, so this is a very busy graph, and it's not, the important thing is actually just that you see trends. So what we measured the total carbon, total organic carbon, we measured the total nitrogen, we measured total organic that we have, and the amount of mineral phosphorus in our samples, which is hydroxyapatite. It is very small amounts, and the importance of that is that if you look at it as a function of mineral phosphorus for the organic carbon, mineral phosphorus, the organic nitrogen, and mineral phosphorus, the organic phosphorus, you see very clear correlations. If you remember, Steffi showed you a Rydfeld ratio of those things. We now done the same thing. So what you see in this plot here is the Rydfeld ratio between the ideal versus our samples. And you see again this very nice link. So what we are showing with this data is that actually the algae seem to biomine a little bit of phosphorus from the minerals for their growth. But this is samples, at the moment I showed you from those big aggregates, what the mineralogy is and what we have also done the geochemistry. But then Steffi has contributed to this paper also, where she looked at the bacterial, the fungal, and the algal community composition in these samples. And again, it's not the habitat which seems to be driving it, it's actually the sampling site. That means, for example, if we sample around here, that is very typical and the same, but actually overall, they are very comparable. But still, we now knew something about the minerals, we knew something about the microbes, but we want to figure out how to put them together. So what we then did is we actually did a couple of experiments with samples which we took from here, and this is now work by Janine McCutcheon and again Chris Williamson, where they did experiments where they took sort of like algal cells and actually fed them nutrients and incubated them for 120 hours and looked at the effect of nitrogen species and phosphorus onto their growth rate, in this particular case expressed as PAR. And it's clearly visible that basically the phosphorus drives this change very dramatically. That means we haven't just measured in the field, but we also done an experiment to see that it's the phosphorus which is driving it. So actually the phosphorus mining is important. But as a geochemist, as a mineralogist, I always ask myself, where does the mineral come from? Because it can either melt out from the ice, it can come from the forefield like I showed you, or it can come from large distances. So in order to do that, we measured the rare earth element composition, and what you see here is yttrium over terbium and lanthanum over samarium. And these blobs show you where the generic African dust would lie from, the Asian dust, and the Greenland samples. And our samples primarily fit into the Greenland samples. We're not excluding potentially a small contribution from Africa and Asia, but it's mostly local. From the data which we have so far, we assume that this was our sampling spot and the samples come from here. And the rocks in front of the glen. So that's one of the works which we are going to do now. So we showed you we have impact of black carbon, impact of microbes, impact of minerals. They all somehow darken the surface. And by looking at these colors, you ask yourself, how on earth can you ground truth something like that at the scale by 25 centimeters, 3 centimeters, and this is 5 centimeters. But if you want to link it to something on a satellite, you have to think about it differently. And Steffi showed one is full reflection in green snow to 0.4, in red snow to 0.45. And in gray ice at the time, we had very few data because we focused, as Steffi mentioned, primarily on the snow. We got down to 0.35. But when we calculated the effect just from the snow, red snow alone, we realized that 13% more melting came from the presence of the red snow. That means you have more melting, more algae grow because they need the water. More algae grow, you lower the albedo, so it's a positive feedback. But still, we didn't know what actually how good these numbers are because we had few numbers. And then part of the black and bloom, this is work from Joe Cook and Andrew Tedstone, where they linked it everything. They measured it on the ground with a much better. We just had a simple radiometer, but now we have a broadband spectrometer. Also to sat measurements. I'll show you some of the data from Joe Cook's paper, which came out in 2020, where he showed that the high algal biomass samples bring down the albedo compared to, let's say, the clean snow from about 0.85, we set up to 0.75, it brings it down, high algal ice algae biomass brings it down to all the way to about 0.35. And he's done that by including a variety of parameters from the mineralogy, from the cell numbers, from cell counts, from pigmentation into a model BioSnicker Go, and done that at the small scale, what we call a pixel of 25 centimeters square, 50 centimeters square. Then linked it to the drone measurements at 250 meters square and 500 meters square pixel, because that is what the satellite sees. And then Andrew Tedstone has taken those data and incorporated them into the bigger scale models where he took satellite data and tried to see how the changes along the Greenland Ice Sheet margin, this is this margin here, how it changed between 2000 and 2016. And these two papers very clearly show that the ice algae alone produce between 13 and 15 more melting, but in some areas up to 25% more melting in localized patches. So it's a huge impact that we have across the Greenland Ice Sheet. Now remember, we only have this edge here, and that's just one part of the Greenland Ice Sheet. And that's where we now basically come in, where I want to mention Alex and Martin, because we have just started beginning of last year, a new project, which is called the Purple, don't ask. It sounds catchy, but this is a ERC funded grant where we want to actually not just look at the two areas where we looked with black and bloom, but we actually want to look in many more areas in green to see where it actually happens everywhere, because we see it from the satellites and actually ground truth that data. So the difficulties is because we now know that it happens, but we actually don't know the mechanisms and the fundamental process that actually trigger, for example, the melting. So in this picture here, what you see is a sort of like a schematic of how a surface looks like of an ice. So this is depth, and this is distance, and this is sort of like a filigrane surface of the ice. To actually measure and quantify the changes in something like that is really, really difficult from the point of view of the ice physics, from the point of view of many, many parameters to measure them. But we aim to measure how we go from snow to slush, to forming snow algae blooms, to go to ice algae blooms, and how these algae bloom emerge, because we don't know what triggers them. We want to also see what happens in winter, because we know that they freeze in. And this is an ice core where you see the snow layer, and you see the ice core, the snow ice interface, and underlying ice with ice algae in here. We don't know what happens to them in winter, and we don't know what triggers the bloom once the melt starts. And in order to do that, we basically will take a bunch of cores, both look at them as they are frozen in the winter using cryo-CT, for example, but we're also looking at the surfaces, again, by preserving these samples during the melt season to see how they developed as a function of time. And then, obviously linked to all this, comparing what we've showed with the snow algae, we're also looking at the whole carbon cycle, because obviously the carbon development in the blooms, what is delivered to the ice, what is utilized on the ice, what is cycled, or what is exported from the ice, how that is linked to the nutrients over the melt season is something which, despite the fact we know it happens, we have still no clue how it happens. We want to differentiate between snow and ice specific signatures, but we want to also look at seasonal and diurnal changes, because these things actually are happening on a daily rotation, but also on the long term. The next step actually have started. And this is a picture from 2019 and 2020, where we were lucky to be able to go in the field, although the weather here wasn't so good. But our goal with Deep Purple is to improve the understanding of this least studied Earth microbiome. But primarily, we want to understand the fundamental controls that glacial algae have on growing around the Greenland Ice Sheet, not just on one place. Because only by combining all these different approaches will we understand and be able to make a contribution to future albedo parameterizations. And actually, the real stars of this work are the people who are actually listed here. And I obviously cannot list all of them. What you see in black, in dark, are all the people who have contributed, for example, here to Black and Bloom, which was basically a NERC-funded and a Helmholtz-funded grant. And these are the PIs. I didn't mention the work of all the students because there was a limitation. And in black at the moment, the students and postdocs which have already contributed to a Deep Purple last year's, and many more which will contribute at the moment. So you think this is over? No, because the future is actually today. Remember, this is the picture from 2019, where we had massive spikes of melting. And again, we had a huge area of Greenland melting in July 2019. And this is not anymore the exception. This will be the norm. And we have to ask ourselves, because if this is the norm, when do we get past the tipping point? Can we stop this? And I look back at the Schellnhuber paper from Nature Climate Change in 2016, where the tipping point for the, let's say, West Antarctic ice sheet and Greenland are linked to the Paris range. And we all say, well, we should not exceed 2. We have exceeded 1.5. We will exceed 1.6. We will exceed 1.7. It doesn't matter. We don't have to just say we don't want to exceed that. Every step counts. And we will get there regardless of how the predictions are. But the important thing is that we need to think about that it was the right climate change, but we need to do something, because otherwise we are willingly shredding our world. And I think that's not the kind of direction we want to go to. And with that, I want to finish up, because I'm saying, we don't just want to save it for us, but primarily I can say that because I'm old, we want to save it for the next generation, and especially all the people who will help us in the next sort of like 5 years and decade to actually solve these problems. And we finish up with Bernie Sanders being with us last year in summer on the ice while we have lunch, and any questions, we're opening up for questions. Thank you very much.
Bernie gets around. It's pretty amazing. For our audience, if you have questions for the speakers, please just pop them into the Q&A. It was a really fascinating talk. I have lots of questions, so I can start while we wait on anyone else. So you talked about the organisms getting biomining phosphorus from the minerals. Where do you think they're getting their nitrogen from?
Steffi, you want to?
Are we talking about the ice algae on the Greenland Ice Sheet now?
Yeah, I mean, are
I guess
fixers or
yeah. So I guess with dust, it's like very, very small traces of nitrogen coming in with dust. On more like mountain glaciers, you can also have inputs from birds or from other animals.
And there is also enough nitrate in the snow itself.
Oh, interesting. Where's the nitrate coming from in the snow? Is it just like seawater? I mean,
we have never seen in the snow itself, we've never seen the fact that there's not enough nitrate. There's always enough nitrate, and depending on where you measure it, the variability is big. But also remember that we at the moment primarily talked about the algae, but we don't know, for example, whether the bacteria are actually, they probably metabolize in a different way. So that means maybe a different kind of nitrogen cycling. And I knew you will ask the question about nitrogen because you are
also nitrogen fixation by bacteria.
Yeah, by bacteria.
Yeah, I was wondering if there was nitrogen fixers, but then they require trace metals. So then you have to have but I can see that there would presumably be a lot of trace metals coming in from the dust as well. That makes oh, that's really interesting.
Yeah, so one of the interesting things was when we started doing this, we measured with ICP-MS trying to measure trace metal concentration. And actually, we had to get new standards because the ice and snow is so clean that in order to measure the ICP-MS for actually the trace metals, we had to literally get a new protocol done because it was cleaner than the normal distilled water that we had. So, but they seem to be not needing a lot. And if you grow these things, actually you don't have to give them lots of food. You actually have to dilute media, because otherwise if it's too much, they don't like it.
Oh, wow. That's so interesting. Yeah.
So our colleagues at the moment in Aarhus, which are actually trying to grow the ice algae, they literally have to dilute by 100-fold at least the normal media, because otherwise they just die. They don't like Christmas. They just like misery.
Extremophiles are so crazy like that. It's amazing. We see this in the Atacama Desert. If you would assume that organisms would be water limited, but if you add water to them, they just completely just explode and die, because they're actually nutrient limited, or they're actually organic carbon limited. So are there also heterotrophs? I mean, is this sort of the base of sort of a microbial ecosystem? Are there heterotrophs that eat the phototrophs?
Yeah, the bacteria heterotrophs, definitely.
Oh.
And then, of course, there's cyanobacteria, the phototrophs.
That's really
But I mean, who is dependent on whom, and how they're all linked? This is all still very much work in progress, like the algal bacterial interactions.
It's really fun.
And there are a couple of PhDs actually drive that at the moment as part of the purple, but it's not, obviously, as you know, it's not trivial.
Rich,
And of course, there are also fungi then, which we shouldn't forget.
Sorry, what was that?
I said, and there are also fungi, which we shouldn't forget, which also play a role in this whole system.
Rich, you said that you would have a question?
Yeah, I have a question about, I think it's something that you're maybe more planning than what you've done with the Deep Purple project, but I was intrigued by the idea that there might be some kind of feedback between the hydrology of the snow, ice, and the biology, and that these things might sort of interact non-linearly, and that they depend on each other. So I wondered if you could say a little bit more about maybe what you know so far, or what you're hoping to, what your hypotheses are for that system.
Oh, you're stretching me. I mean, I wish Ian or Joe would be here, because they are our hydrologists. And so basically try to look at the ice physics is something really difficult, because trying to look at how a surface of the ice melts as a function of time is difficult, because on the ice, for example, the minerals and the microbes are actually forming almost like a very small thin layer. But because it's constantly changing, even just measuring something like ice density or particle size is not non-trivial. So if you remember that picture that I showed you, that of that very filigrane surface, to measure, to model, to actually analyze that, then actually introduce it into a model, I wouldn't want to do that, but that's Joe Cook and Ian will do that. And the people from the weathering crust around Martin Tranter in Deep Purple, they will do that. And hopefully we can solve it. We will help with measuring the biology, measuring the 3D distribution and the 3D arrangement of these things, because we know that they form a very thin biofilm, but because they change all the time, we don't know at the small scale and how to actually scale something up. Because if you measure something in a microscope and then you want to bring it up to 25 microns, or sorry, centimeters to 500 meters and so on, it's really difficult because of that heterogeneity. But it only works by having all these people working together and asking almost the same questions from 17 different directions. And maybe we'll get an answer that way by working together. I'm not sure that answers your question, but I wouldn't even know how to start with answering a question about hydrology, because that's like...
Okay, so maybe I was unfair in making it too much about hydrology, but I guess I'm just wondering if you envision a feedback between the biology and the presence of water, such that the presence of water promotes growth, the growth reduces albedo, promotes melting, and therefore you get some runaway or
Yes.
Some self-reinforcing feedback. Is that...
Yes, that is correct. But the question is how to quantify exactly that measure. We know it, like I said, we know it happens. And part of Deep Purple is to try to actually quantify that process and the mechanism that actually triggers it. But also, how does it change during a melt season? Because remember, at the beginning, when you have snow cover, and once the snow gone, you get this slush slush regime, but we don't know whether the slush regime and what happens underneath the slush, underneath the snow. So how is that all linked? And that's part of the hydrological process that we have, problem that we're having, and hope somebody will solve it.
Thank you.
There's a question from Laura Perini, who is asking about how the algae get the pee. Do the algae scavenge the phosphorus themselves? And if so, are they releasing enzymes, or how do you think they're doing that? Or do they get help from other microorganisms or fungi?
Thanks, Laura. I guess the problem is we don't know the answer yet. And although we've done some initial experiments, both, like I showed you some experiments on the ice that we've done, where we fed them not just the dissolved phosphorus, but we also try to do it with mineral phosphorus. But I think those are the kind of experiments which we'll try to do now, both with cultured snow algae, but we don't yet have the cultured ice algae to actually see whether they produce particular exudates or whether they eat particular part of the carbons. So that's part of the experimental system that we are looking for in the next couple of years.
Sorry, Liane, you sort of blanked out at the end there. Could you repeat that last bit?
Oh, so I don't know how far you got, but actually we will do in the next couple of years. We have a bunch of incubators, both in Aarhus and in GFZ, where we actually do experiments once we have the snow algae and the ice algae in culture to actually look exactly at that. Whether are there particular types of the mineralogy? Is it particular particle size? Is it particular enzymes that they actually produce? Or is it particular chemical components in the EPS, for example, that they need because they produce a lot of biofilms? I showed you one picture with the biofilms. Where is it the biofilm part which actually makes the attachment in aggregation and the extraction of the phosphorus faster or slower? At the moment, I'm just basically spouting potential nonsense, but we don't know because we have to actually do those experiments. But because we don't yet have the ice algae in culture, it's really difficult. Remember, that's one of the things, most people think the ice algae are in culture, but they are not yet in culture. We're just working on that. And once they're in culture, we can do, like Steffi said, we don't have the genomes for the snow algae yet, but we don't have the genomes for the ice algae yet either. Once we have those, we can also see, do they have particular genes that they need, or do they actually are active if you put them in with phosphorus or not? So I think you can do all of those experiments once we have them.
Just sort of a follow-up.
That you want to add?
No, but just as you said, it's all very much at the very beginning. I mean, like in other environments, you have all these microbes in culture or the reference genomes, and with the glacial microbiology, you just start from scratch, basically.
And it sounds like, you know, we gave a talk about an hour about something which we know, but actually I hope we also showed you that we just literally scratched the surface, and we actually know, we know it happens. We don't know yet how it happens, what the mechanisms are.
Just a brief follow-up. Oh, okay. There's another question, actually, so I'll go ahead and read that one. Ava Doting wants to know, Steffi, regarding the metabolites found in red-green snow, is the bacterial abundance so low that bacterial metabolites are unlikely to interfere here, or do the data represent the combined metabolome? And she says, thanks for a great presentation.
Thank you. Yes, it is combined, and yes, the algal abundance is higher than the bacterial, and yes, we assume that the algal contribution is higher than the bacterial. We did not manage to separate the algal from the bacterial, but then also, like, when we searched the metabolites and in which organisms they appear, I mean, they were mainly like found in other algae. So of course, it's again a lot of assuming, and ideally in the future we will be able to separate the algal contribution from the bacteria. But when looking at abundances and like total biomass, we assume and hope that most of it actually comes from the algae, because they are so dominant in these green and red rooms.
Do they grow really slowly? I mean, is that part of the problem with culturing them?
Actually, once you get these green blooms with the young snow algal cells, they can really grow within a few days. They kind of like explode. The tricky bit to grow them in the lab is to find out what they actually need, because they don't need a lot, but they need something. And also the big issue is you can't really grow them in snow or ice in the lab. So it's like the physical state of the medium. They're not happy in liquid media, and they're not happy on like algal plates. So they need a little bit of liquid water, but not too much. And it's really tricky to simulate in a lab. And once we tried with artificial snow, that didn't work. And we tried all kinds of things. And also other groups who are experts in growing them, they're really struggling.
So, I mean, you have to remember that the red snow algae are used a lot for food production, because they actually produce a pigment which makes your salmon pink. Your salmon would be normally, the farmed salmon would be normally white, but the astaxanthin, which is produced by both the cryophilic and the mesophilic equivalent of these algae, is actually what produces astaxanthin, which is the pigment which they feed to salmon to make it pink, because otherwise you wouldn't like your salmon.
Has to look nice.
People have managed to put them in culture, but it's not at the conditions that we have them on the ice. So it's always a sort of like a compromise that you make.
It's also in these red snow algal blooms, they're often made up of spores. And when people try to bring those spores back in the lab and try to make them germinate, it's just impossible. They just rest and don't do anything.
I'd like to say I would like my salmon more if it wasn't pink, but
good on you.
Can start a petition.
Non-farmed salmon is pink by nature because it actually has that part of the metabolic pathway, but farmed salmon, because they don't move enough, they need to be fed astaxanthin to go pink.
Amazing. Are there any other questions from the audience? If not, I will thank Liane and Stefanie again for a really fascinating talk. And thanks to everyone who is attending, both on Zoom and on YouTube. I'll remind you all to please check our webpage and Twitter feed to keep track of upcoming seminars. And it looks like our next talk will be on the 22nd of April, featuring Professor Isabel Montañez from UC Davis and Dr. Sophia Makarowicz from the University of Michigan. So I hope to see everyone there.
Thank you very much for the invitation.
Thank you.
Thank you. It was really great. Bye-bye.
Bye.